Biological Safety Training, Chulalongkorn Research Institute. The principal characteristics of a biological agent that determines its biohazard potential are its capability to infect and cause disease in a susceptible human, its virulence as measured by the severity of disease, and the availability of preventive measures and effective treatments for the disease. Biohazardous agents include microorganisms, allergens and toxins derived from microorganisms, allergens and toxins derived from plants or animals. Why practice biosafety? A fundamental objective of the biosafety program is the containment of potentially harmful biological agents in order to reduce or eliminate exposure of laboratory workers and other persons, and prevent the release of biohazardous agents to the environment. The principle of biosafety. The principle of biosafety involves the application of a combination of specific laboratory practices and procedures, laboratory facilities, and safety equipment when working with hazardous biological agents. Proper laboratory practice and equipment prevent exposure to hazardous biological agents. The proper use of personal protective equipment, such as gloves, lab coats, and eye protection, can significantly reduce risk from exposure to potentially infectious materials. Vaccines may provide an increased level of personal protection. Disease surveillance will decrease the harm caused by such infections. Lastly, risk assessment of the work to be done with a specific agent will determine the appropriate combination of these elements. Can you recognize unsafe laboratory practices? What is wrong in these pictures? Do you know your lab's biosafety level? What are the proper procedures and practices for your biosafety level? How does waste need to be decontaminated at your biosafety level? The term biosafety level refers to the level of the biocontainment precautions required to contain dangerous biological agents. The different biosafety levels developed for microbiological and biomedical laboratories provide increased levels of personnel and environmental protection. The guidelines for each biosafety level work from the premise that safe work sites result from a combination of laboratory practices, techniques, safety equipment, and proper laboratory facilities. The practices, safety equipment, and facility design and construction of a biosafety level 1 or BSL-1 facility are appropriate for laboratories in which work is performed using defined and well-characterized strains of viable microorganisms that are not known to consistently cause disease in healthy adult humans. For example, Bacillus subtilis and Escherichia coli. Biosafety Level 2 practices, equipment, and facility design and construction are applicable to clinical, diagnostic, teaching, and other laboratories in which work is performed using a broad spectrum of indigenous moderate-risk agents that are present in the community and are associated with human disease of varying severity. Examples of agents that should be handled in a BSL-2 laboratory are hepatitis B virus, certain other blood-borne pathogens, salmonella, human body fluids, and cell lines. Biosafety Level 3 practices, safety equipment, and facility design and construction are applicable to clinical, diagnostic, teaching, research, or production facilities in which work is performed using indigenous or exotic agents with the capacity for respiratory transmission that may cause serious and potentially lethal infections. Examples of agents that should be manipulated in a BSL-3 facility are Mycobacteria tuberculosis and H1N1 influenza virus. Biosafety Level 4 practices, safety equipment, and facility design and construction are applicable to facilities that work with dangerous and exotic agents that pose a high individual risk of aerosol-transmitted laboratory infections and life-threatening disease for which there is no applicable vaccine or therapy. Currently, only BSL-1 and BSL-2 work is being done at CRI. Standard microbiological practices. Standard practices and safety equipment for BSL-1 and 2. BSL-1 represents a basic level of containment that relies on standard microbiological practices. Hand washing is one of the most important procedures that can be used by laboratory workers to prevent the spread of microbiological agents, radioactive materials, or chemicals from the laboratory environment. Persons must wash their hands after working with hazardous materials and before leaving the laboratory. Use of liquid soap is generally preferable to bar soap. 20 seconds of vigorous lathering will remove most contamination very effectively. After drying your hands with a paper towel, you can use the towel to turn off the faucet and thus avoid recontaminating your hands. Use eye and face protection to minimize the chance of facial contamination via splashes or aerosols. Decontaminate work surfaces before and after completion of work, as well as after any spill of potentially infectious material with an appropriate disinfectant. A sign incorporating the universal biohazard symbol must be posted at the entrance to the laboratory where infectious agents are present. Procedures for the safe handling of sharps, such as needles, scalpels, pipettes, and broken glassware, must be in place. Decontaminate or sterilize all cultures, stocks, and other potentially infectious materials before disposal. Keep the laboratory neat and organized. Don't. Eating, drinking, smoking, handling contact lenses, applying cosmetics, and storing food for human consumption must not be permitted in laboratory areas. Food must be stored outside the laboratory area in cabinets or refrigerators designated and used for this purpose only. Mouth pipetting is prohibited. Mechanical pipetting devices must be used. No sandals. Shoes must completely cover the foot. Do not touch clean surfaces with gloved hands. Gloves should be removed before leaving the laboratory area. Personal protective equipment. For safety, persons working in the lab should wear a lab coat to prevent contamination of personal clothing. Gloves must be worn while working to protect hands from exposure to hazardous materials. Additional protective equipment may include working behind a splatter shield or wearing eye or face protection when conducting procedures that have the potential to create splashes of microorganisms or other hazardous materials. Persons who wear contact lenses in the laboratory should also wear eye protection. Long hair should be tied back. As the biosafety level increases, all those microbiological practices and procedures delineated for the lower levels are carried forward to the next higher level. Thus, the standard microbiological practices required at BSL-1 are still in effect at BSL-2, with emphasis on wearing gloves, using mechanical pipetting devices, and attention to handling sharps. In BSL-2 laboratories, some work may be done on the open bench by persons wearing the appropriate protective clothing or gear. Any work that may produce splatters or aerosols of infectious materials should be performed inside a biological safety cabinet, or BSC. Other containment devices, such as aerosol containment centrifuge cups, should be used where appropriate. Special practices. In most situations, it is best to use single-use needles and syringes. Do not break, bend, or recap needles. Needles and syringes, butterfly needles and their associated tubing, and similar devices should be discarded intact into a puncture and leak-proof container. Other sharps items, such as broken glass, should not be picked up by hand. All persons entering the laboratory must be advised of the potential hazards and meet specific entry and exit requirements. Workers should be familiar with the biosafety manual and should receive proper training with annual updates. Biohazard signs should be posted on all entry doors in the laboratory. Other special practices include the decontamination of work surfaces after completing work with infectious materials, keeping non-research animals out of the laboratory, and reporting spills and accidents. An accident logbook is a useful means for recording events that have gone wrong. It is important to document these events, not for punitive action, but to be able to better understand what happened with an eye to preventing similar events in the future. Safety equipment, personal protective equipment. Personal protective equipment for BSL-2 is the same as that for BSL-1. Gloves and lab coats must be worn, and eye protection should be used when splashes or aerosols are anticipated. Safety equipment, primary barrier. At BSL-2, all work that might create aerosols of infectious materials should be done using some form of containment. The most common device for this is the biological safety cabinet. The most common cabinet in use is referred to as a Class II Type A biosafety cabinet. Operational practices. Gloves. The correct use of gloves will prevent the spread of infection. You should remove your gloves when a procedure has been completed, if the glove is torn, and before touching clean objects such as doorknobs and telephones. Gloving technique. When you remove your gloves, your intent is to avoid touching the contaminated surface of the gloves with your bare hands. When taking off your gloves, firmly grip one glove at the base of the palm and pull it off inside out. Holding the balled-up glove in the palm of your other gloved hand, slip your bare fingers under the wrist of the remaining glove without touching its outer surface. Push the glove down and off with the first glove inside of it. One glove is now inside the other, and both are inside out. Properly dispose of the used rubber gloves. Traveling from room to room. It is fairly routine that personnel travel from room to room. You have two options. Option A, place your sample in a tray or container, remove your gloves, and wash your hands. Move to the new destination, put on new gloves, open the container, and proceed with your experiment. Option B, you may use the one glove technique. Remove one glove and use your ungloved hand to open doors and manipulate controls on equipment. Keep your gloved hand away from common surfaces and use it to handle the specimen container only. How to deal with spills. Cleaning up small biological spills in a containment device. When you clean up small biological spills in a contained space, such as a biological safety cabinet or centrifuge, carefully follow these steps. First, stop work and carefully cover the spill with paper towels, starting from the outside of the spill and working inward toward the center. Then, gently apply an appropriate decontamination solution to the paper towels. Allow the solution to contact the spill for at least 15 minutes. Longer if spore-forming organisms are involved. If there is any broken glass, pick it up with forceps and place it in a sharps container. Next, gently pick up the paper towels with tongs and place the paper towels in a waste bag for further decontamination. The spill area should then be cleaned again using the decontamination agent. Contaminated gloves should be disposed of properly. In the event of a large spill outside the biological cabinet, a biological spill kit should be used. Follow the instructions provided with the kit. Waste management. Many biohazardous materials will grow if left to their own devices. For this reason, biological wastes should be disposed of quickly and shouldn't be kept around for more than a few days. At CRI, biohazardous waste is either autoclaved on site or treated with disinfectant on site before being discarded in the general trash, or discarded in the biological hazard trash to be picked up by a contractor and incinerated. The proper use of personal protective equipment can significantly reduce your risk from exposure to potentially infectious materials. Remember that even if other safety controls are employed in your facility to eliminate or minimize biohazards, personal protective equipment remains your last line of defense against potential exposures. The effectiveness of that defense is literally in your hands.